Um, so thank you. Hi everyone, and thank you the conference organizers. So give me the opportunity to talk today. So my name is Hao. I'm from IMIT University. From Australia. So my research and my group research is on Campylobacter hepaticus. This is a bacteria which cause spotty liver disease in chickens. And we started this research 5-6 years ago, and then we made a lot of progress on this area, which I'm going to show you. But firstly, I'm going to talk about SLD first. What is SLD, spotty liver disease in chicken, because some of you may not heard about it. So spotty liver disease in chickens is a significant disease in poultry industry, especially in Australia, because it can cause mortality and also the egg production reduction as well. So in 1950, was first reported in the USA, and then it was reported in other countries. But about 20 years ago, it came back, and then now it's called problem for Australian poultry industry. And then it's also reported in other countries, which I'm going to talk about it later. So what did we do? So the disease was reported 60 years ago, but the cause of the disease still unknown. Until 2016, a group in the UK, Grosso, they isolated a new bacteria from SLD chickens, infected chickens, but they didn't go further to identify. And then shortly after that, our group had isolated the bacteria, new bacteria, and then we went further, and then we did all the biochemical test, and we did all the genome sequencing, and we can conclude this is a new bacteria, which we named Campylobacter hepaticus, and the type strain is HP10. So hepaticus means that it was isolated from liver. It's hepaticus means liver. And then we publish. Then we also study, and then we put it in chickens, and then we also produce induction model, which means that we put in chickens, and then they also reproduce disease in layer hen, and then the model had been used later on. So what's known about his C. hepaticus, Campylobacter, the bacteria we call spotty liver disease in chicken? We test in the LMH cells, it means the liver cells, chicken liver cells, which is highly invasive compared to other Campylobacter, which you may know other Campylobacter such as Campylobacter jejuni, Campylobacter coli, which they call food poisoning in human. And we also develop endpoint PCR and quantitative PCR to detect this bacteria. And we published, and other group have used this method. So we have the rapid method to differentiate as well with other Campylobacter. And then we also have the endpoint PCR to detect this bacteria. And then we also optimize growth of C. hepaticus, but I'm going to show it later. And then we also do the genomics and pathogenicity studies of hepaticus. So what makes this one different from other Campylobacter? So for the cysteine phylogenetic tree, as you can see, after do the cysteine, we made the tree and to compare with other Campylobacter. And as you can see in here, they group together Campylobacter hepaticus, and they different from other Campylobacter. And we also did the comparative genomics. And so Campylobacter hepaticus, they form a separate clade compared to other Campylobacter. Like closest is Campylobacter jejuni and Campylobacter coli. But at Campylobacter jejuni and Campylobacter coli, they call disease in human, but normally they are they're not calling any disease in chickens. But in contrast, Campylobacter hepaticus, they call disease in chickens, but in layer bird. It means that the bird would give eggs. So, but the hepaticus haven't been reported like call disease in human, but we haven't tested, but that's what we think. So we did the tree, and we collected sample from different states. Victoria, South Australia, Queensland, New South Wales, and all states in Australia and Queensland. So they form a separate clade here. They group together, and then we also have the genome from the UK. So we compare, and now we also have the genome from the US as well, but we haven't included here yet. So when we did the proteome comparisons, as you can see, most of them, they are closely similar. They are very similar within the purple color, but there are the strains from Queensland. They are quite different. With, like you see, is lower percentage, like similarity percentage. So when we did the genome sequencing, you also found the plasmid, which contain TET-O gene, which you can know that TET-O is responsible for tetracycline resistant. And so we had the report that from one of the states, the disease at the moment, they have to use antibiotics to treat the disease. And in one of the states, they report that the chickens didn't respond to the treatment. So we think that maybe because the tetracycline resistant plasmid is make it resistant to the antibiotics used. And then we did animal trials to compare the decrease of virulence of different isolates. And as you can see in here, they have different. They went the bird challenge with the same dose. The one of the strain, they call more disease than other, but all of them can cause disease as well. We also did the pan-genome to compare, to see the virulent factors, and then we can see that there are 4 chemotoxic proteins with the homology is less than 8% to other Campylobacter. So this may have this bacteria with species can move to the liver because it's called the disease in the liver, but this needs to be confirmed in the lab. And then we also found that liposaccharide locus, the rearrangement, which I'm going to show later, like the rearrangement. They have the glucose utilization metabolism operon in here as well. And then, but normally Campylobacter cannot use the glucose, but this is Campylobacter is different. So they have some changes in their genome which make these differences. And we also did RNA sequencing to compare transcriptomic. So we compare between the one in vitro conditions. It means that we also grow them in the lab, and also in vitro, we put them in chickens, and then we compare the hepaticus with grow in the lab and with grow in chicken, and to see whether any gene is spread more in the in vitro compared to in vivo compared to in vitro, whether it's contribute to the virulence or not. So we did RNA isolation sequencing, and then we did the differential expression analysis of the 2 conditions. And as you can see here, some gene up and down regulated with the, for example, fatty acids, lipid, they up and regulated. So they may contribute to the pathogenicity of the Campylobacter hepaticus. So, for example, I listed in here. So we found the upregulation of copper transporting ATP genes, whether they have the role in copper export or not, or they also have PHB metabolite genes as well, whether they thanks to this gene upregulated, they can respond well to the limited nutrients in bile that make them can survive in bile. They also and then we also found other interesting things as well. And then, but interestingly, we found that downregulation of many flagella and chemotoxic genes with because flagella also contribute to pathogenicity as well. But we found that is in general we find in this bacteria. I also would like to talk about our latest findings on SLD. So we have a project to study the epidemiology study of SLD. And then we collected a lot of samples from different farms. And then by accident, we detect a new species, Campylobacter hepaticus. Why is by accident? Because we had the PISA to detect, and then the PISA also positive. So we didn't know that it is a new species. But then we did the genome sequencing, and then when we did assembly, like at the routine one, and then we found the genome that is different. They have bigger GC, higher GC content. So hepaticus have only 28%. This one have 30%. And then we did other tests to confirm that it's a new species. So the same species causing the same, different species causing the same problems. The same disease, but like human as well. Campylobacter jejuni and Campylobacter coli, they are different species, but they both cause disease in human. We also made progress on improve culture conditions for growth of Campylobacter hepaticus as well, which I'm going to talk about it later. And we also demonstrated that C. hepaticus can enter a viable but non-culturable state. And then we also use experimental disease induction models XLD, which I'm going to present later. So what's new recently about SLD research? So last year, so we detected in Australia like a few years ago, 5 years ago for hepaticus. For B-list, we detected last year. And then USA 2 years ago, there's a paper also describe they detected, and UK they detected like 2015, and then other country also just detected, just reported as well. New Zealand also published, and Jordan, recently Jordan, and I have students from Vietnam, and then they also found T. hepaticus in spotty liver disease, but they haven't confirmed yet. But that's what we know. So what's about the new species C. Campylobacter bilis? So as I said, it's identified during culturing and whole genome sequencing. So they closely related to C. hepaticus. But based on phenotype, like we did a lot of biochemical tests, and then also genomic DNA, we compared the genome, and we found that it is the new species, which we only detect in 2 states. And normally it's hepaticus if we detect if the chicken has SLD disease. And then we put in the bird, and then we found the disease, confirmed that it's a cause of the disease. And then we put the time strain, we already submitted the time strain to ATCC and NCPC. And then the paper, international, we submit the paper to International Journal of Systematic and Evolutionary Microbiology, and then it's in progress. Hopefully it will be published soon. And then we also improve culturing of C. hepaticus. This is a big progress. So our student, PhD student, he developed a meter with a liquid meter. So normally, like a few years ago, when we did do the trial, we have to grow them on the agar plate, because these fattidious bacteria, they're very hard to grow. But it takes a lot of time. For example, because if we work on one chicken, we need one place. If we do the trial on 100 chickens, we have to grow a lot of place. And then because this bacteria is very sensitive, and then very prone to contamination, so we have to grow a much more than that. You could not believe how many places we have to use for each trial. But since the discovery of the liquid culturing method, it makes our life easier. So we put different additives to boost blood growth, and then make it 10-fold higher growth, which is very significant. So we can make from 10 to the 8, 3 10 to the 8 to 3 10 to the 9, which is enough for to put in chicken. And then we put in chicken, and then it's also produce disease as well. so they can produce a disease. And then we already published in a scientific report we just published recently, last month, about the method. And then we use this for our following animal trials, like to study the vaccine for this bacteria. And then this student also work on the to see whether this one can enter viable but non-culturable states. It means that they are there, but they're not dead. They're still there, but they survive. And then when the condition is good, they can come back and thrive. So we found that the B, and C cells are coccoiform. And then when the condition is good, they come back and then can grow. And you can see in here for 65 days, you can see they still alive. So this can explain why we can see they still survive in the farm, even after the farm. From the farm, they can be transferred from farm to farm as well, or even in the very harsh environment. The birds still can have spotty liver disease, but disease bacteria in the lab, they are very hard to grow. So our other SLD progress, that we another student, she developed ELISA assays, which now, so before we develop PISA assay, we detect the DNA of the bacteria. So if we collect sample, we detect the DNA. We don't know that whether this is a current infection or past infection. But for ELISA assay, then we can. So this assay, you use the we use the serum. We check whether their blood whether contain anti-C hepaticus. Then we know that whether it is a past infection or current infection. If you use both. So we can then, so ELISA can use to see the previously exposed to hepaticus, because if it's already previously exposed, the PISA cannot be detected, and then we can see the IgG responses. So there's a paper also have been published in Avon Pathology. And then we start a new AIC linkage project. It is the time of the government funding from Australia, with other company, and also the London School of Hygiene in UK. So we developed the vaccine against CG29, and we hope that it can work in hepaticus as well, which based on complex and glycan surface carbohydrate. So I would like to acknowledge my team, Professor Rob Moore, and also other members of the lab from MIT, Ben, Kan, student Kan, and Chitra, and also our industry partner. They collected samples for us, and then we also did the trial together. Scholars like Peter Scott, Arif, and Tyrone team, and then other uni, Melbourne Uni, for the bioinformatics together. And thank you for funding from Poultry Hub. We received several funding from Poultry Hub for our study, and also Australian eggs to study the toxins produced in Campylobacter hepaticus, and also the poultry CRC. And thank you for listening.